us that membranes are movable light, right? So remember we started with membranes in chapter 13, going over, kind of what they can do in terms of the molecules and signals, at least in terms of action potentials. But there are other ways that we can get signals into the cell, and of course these have to be moved through the membrane, right? So how do these signals get through the membrane? So signal transduction, right, describes how the cell responds to external stimuli, right? So how are these signals going to be responded to by the cell? And these have to be responded to by the cell because, of course, one of the characteristics of living systems is that they have to respond to external stimuli. All right. So we have some types of receptors that we're going to talk about in depth today. For example, the epinephrine receptor, the insulin receptor, and the epidermal growth factor receptor, right? So we're going to focus a lot on the adrenergic receptors, how adrenaline or epinephrine actually works to accomplish its goals. Remember, epinephrine, also known as adrenaline, is the fight or flight hormone, right? So how does this lead to different activating signals in the cell in order to either get ready to fight or get ready to run away? And the same with insulin, right? Insulin also, right after we have a big meal, insulin is released, and that causes the uptake of glucose. How does insulin cause that to happen? Well, these all happen via signaling transduction cascades, or signaling cascades, where we have a signal. This leads to reception, obviously, by the receptor, whether it's the adrenergic receptor, the insulin receptor, glucagon receptor, or so on. And between reception and transduction, there's this time of amplification. What this means is a very small signal can lead to a very big output, kind of like a transistor, right? So a transistor, of course, I don't know about of course, but a transistor is essentially just a very simple amplifier, okay? And this is what these receptors are going to do. So one, for example, glucagon molecule or epinephrine molecule can lead to the activation of many thousands of downstream targets, even from one single binding event. That's transduction. Of course, that leads to responses. The responses depend on which receptor was activated. And which receptor is activated depends on which receptor is expressed by the cell. So different tissue types will express different receptors depending on what that cell is supposed to be doing. So for example, the heart is going to express a different complement of receptors for adrenaline compared to smooth muscle tissue, right? Because they have to respond to the same signal in different ways. Right? We'll see some of those today. Now, when we bind our external molecule, again, for example, epinephrine or glucagon, insulin, things like this, okay, this leads to the production of what we call secondary messengers inside the cell. So we have our primary messenger, which is the signal itself, and this leads to the production of secondary messengers in the cell. Common secondary messengers, ones that we're going to talk about today, are cyclic AMP, cyclic GMP, right? So notice here this is just an adenosine, right? But the phosphate on the 5' carbon is looped back around, connecting to the 3' carbon. So we have a phosphodiester bond here internally. We also produce calcium ions in the phosphodienostatide cascade. We have inositol 1,4,5 trisphosphate, IP3, and diacylglycerol, right? So actually all these three work together in the phosphodienostatide cascade. Cyclic AMP works in the adrenergic cascade. Cyclic GMP, we'll see, works in the G-alpha, or no, actually works in olfaction, as we'll see in just a second here. Actually, all right, so main class of cell surface receptors belong to this 7TM helix receptor family. We've talked about 7TM receptors before. There are 7 transmembrane helices. They each span the membrane, and a large class of molecules, or of receptors rather, belong to the 7TM family. Most notably, right, the adrenergic receptor, olfactory receptors, and so on. These mediate many biological functions, right? Specifically smell, taste, vision, okay, hormone action, hormone secretion, and neurotransmission, right? And this also relates, of course, to smell, taste, and vision. Okay, so these 7TM family of receptors are actually quite important, right? So let's start with the epinephrine receptor, right? So the epinephrine receptor, again, belongs to the 7TM family, and we see, again, is that each of these helices are spanning the membrane. Here we have just 2 other 7TM receptors. We have for rhodopsin, which we use vision, which we'll talk about later on, and we have the adrenergic receptors we're looking at here, specifically the beta-2 adrenergic receptor, right? It's different than the alpha adrenergic receptor, which we'll talk about later on. But here, the beta adrenergic receptor, again, binds epinephrine on the extracellular side of the membrane, okay? And this binding event has to trigger something in the receptor to signal to inside the cells, right? And that's exactly what happens, right? Upon epinephrine binding, something changes in the structure of the receptor here. And this causes these three, what we call G proteins, to also change what they do, right? We have three G proteins, G alpha here, purple, G beta, and G gamma in yellow here. And what happens upon epinephrine binding is that the affinity between G alpha and G beta goes away, right? So G beta and G gamma remain associated, but they just flow off into nowhere. They remain attached to the membrane. They're they're anchored rather, membrane proteins, and so is G alpha. Now, once we have the release of G beta gamma from G alpha, this GDP, which is always present in G alpha, is now exchanged for a GTP. So we lose the GDP here and we gain the GTP, and G alpha is now free to diffuse laterally through the membrane until it encounters another integral membrane protein, adenylate cyclase. When G alpha, which we now call G alpha S for stimulated, when this GTP bound G alpha encounters this integral membrane adenylate cyclase, right, this activates it. And what adenylate cyclase does is it converts ATP into cyclic AMP, that secondary messenger that we saw over here. Okay? So now we have the production of cyclic AMP. Cyclic AMP then goes on and binds to protein kinase A, activating it. Protein kinase A is, as the name implies, a kinase. And what protein kinase A, or PKA, is going to do is going to phosphorylate many downstream targets, either activating it or inactivating it. So in the case of the beta adrenergic receptor, where it's responding to epinephrine, in cells that are responsible for, say, storing energy, right? Well, what is epinephrine signaling the cell to do? Well, it's signaling that energy is going to be needed right away. So what PKA is going to do is it's going to break down its carbohydrate stores, luckily in the form of glycogen. It's going to break those down into glucose so that we can enter glycolysis and generate energy that way. At the same time, PKA is going to turn off the mechanisms which store glucose, right? So the whole point here is that we are mobilizing glucose so that we can use it for energy, because that's what epinephrine is signaling to that particular tissue, okay? Now, in this whole setup here, right, we go back a few slides here. Remember, we have to have these levels of amplification, right? Here, we're seeing two levels of amplification right off the bat, right? Once we get one single binding event, one epinephrine binding to one beta adrenergic receptor, this beta adrenergic receptor is capable of activating many G alphas, okay? So it'll activate the G alpha that we see here. Once these dissociate, G beta gamma goes off and diffuses laterally. G alpha goes off and activates adenylate cyclase. This leaves this 17M receptor, the beta AR receptor, in its active form because it's still bound to epinephrine. So we can find another G alpha beta gamma trimer and activate that. Once those are activated, it can activate another one and another one and another one. So one binding event has led to the activation of many G alphas, which lead to the activation of many adenylate cyclases. This is not a level of amplification because there's a 1-to-1 ratio between an activated G alpha and an activated adenylate cyclase. However, activated adenylate cyclase is able to generate many cyclic AMPs, right? So it continues to generate cyclic AMP as long as G alpha remains attached. So there is our second level of amplification. So we have many G alphas that are activated, which activate adenylate cyclase, produce many cyclic AMPs for each binding event between G alpha and adenylate cyclase. Our last level of amplification is here at protein kinase A. There is a 4-to-1 ratio between the number of cyclic AMP to the number of active protein kinase A that are generated, but each active protein kinase A can now phosphorylate many downstream targets. So we have a total of 3 levels of amplification here from one single binding event. Right? So we can imagine, even if you pick very small numbers, say a beta AR receptor can activate, 100 G alphas, right, which activates 100 adenylate cyclases. Now each of these adenylate cyclases can produce, let's say, 1,000 cyclic AMPs. Well, now we're up to 100,000 cyclic AMPs, which can activate, it's 100,000 divided by 4, 125,000 protein kinase A molecules, and these protein kinase A molecules can activate many thousands of downstream targets, right? So now we're back up to close to 100,000 activated targets, all from one single binding event, right? So we see why that amplification is so important. Now this G beta gamma that is released upon activation by beta adrenergic receptor is not, just garbage. This G beta gamma can also participate in signaling. For example, in the heart, okay, this G beta gamma can actually interact with calcium channels. And these calcium channels allow the movement of calcium into the cell, right? And what do we know about having calcium in the cytosol of a muscle? What does that trigger? It triggers muscle contraction, right? So in this way, when we activate, right, the beta adrenergic receptor in the heart tissues specifically, what happens? All right, well, let's think. We activate protein kinase A, like we went over here, right? So we go through this whole signaling cascade, activating protein kinase A, and protein kinase A acts to liberate energy, right? So we are liberating glucose, mobilizing glucose in order to make more energy, and we are also importing calcium into the cytosol, which is going to make it easier for the cell to use that energy to contract, right? And of course, this is what we want in the presence of adrenaline. We're about to fight something really big or run really fast. So we need a lot of energy, and we need to be able to contract the heart muscles very efficiently. All right. I already talked about this. I heard myself here. Yes, G alpha S uses the G beta gamma interface to interact with the adenylate cyclase, which is why, right, we come back here, right, which is why G alpha here is not able to activate adenylate cyclase, because this face of it here, this interface, is in the G beta gamma interface, right? Once these dissociate, now this face is free to interact with adenylate cyclase and activate it. Right? Very good. All right, now cyclic AMP acts to activate protein kinase A. Protein kinase A is activated by the release of the regulatory subunits, right? So let's look at the structure of protein kinase A. It's a heterotetramer, right? So we have two catalytic subunits and two regulatory subunits. The regulatory subunits have what we call a pseudo-substrate sequence. Okay, well, what does this mean? Well, any kinase is going to have a consensus sequence. That is, a kinase phosphorylates a protein, but where does a specific kinase phosphorylate a specific protein? Well, it looks for a certain sequence of amino acids in that protein that it can attach to and then transfer the phosphorylated ATP onto that target protein. That's the consensus sequence. In the case of protein kinase A, the sequence that it's going to recognize is RRGSI, right? So two arginines, a glycine, a serine, and an isoleucine. Right? If it sees that on a protein, it will transfer a phosphorylate from ATP onto this serine here, generating phosphoserine. And this will either activate that specific protein or inactivate that specific protein, depending on which pathway it's a part of. A pseudo-substrate sequence is a sequence that resembles the consensus sequence, but is different enough that it can't be acted on. And the way that this pseudo-substrate sequence is structured, it's got this RRG, but in the place of serine, it's got an alanine, and then followed by isoleucine, so it's RRGAI. Alanine cannot be phosphorylated, so while it looks very similar to RRGSI, the consensus sequence, it's just missing the hydroxyl on that methyl group of the alanine, okay? It can bind the active site of the catalytic subunits, but it cannot be acted on. So effectively what's going on here is that the catalytic subunits keep trying and trying to phosphorylate this, because it's recognizing this as the correct shape, the correct structure that it should be able to phosphorylate, but due to the structure of the alanine, it can't do anything with it. So this catalytic subunit is just kept busy permanently. Now, 4 cyclic AMP molecules, 2 for each regulatory subunit, bind. And when we get the binding of these cyclic AMPs, this changes the structure of the pseudo-substrate sequence enough that now it doesn't look like the consensus sequence anymore. The actual structure is too different upon binding cyclic A and P. So they fall out of the active site, releasing the catalytic subunits, but now go on and find actual consensus sites on actual target proteins and phosphorylate them. And these remain bound together in this regulatory dimer, right? Eventually, these cyclic A and Ps dissociate, right? Remember, whenever we have binding between two things, there's always a KD. So this is not a permanent binding event. And when these fall off, okay, the action of cyclic A and P phosphodiesterase, which is universally present, will start to degrade the cyclic A and P. When this is now no longer bound to cyclic A and P, we revert back to these catalytic subunits finding the pseudo-substrate sequence and being inactivated. Right, so we can see how the production of cyclic A and P by adenylate cyclase leads mechanistically to the activation of protein kinase A, which then, of course, goes on and phosphorylates downstream targets, which again either activates them or inactivates them, depending on which pathway we find them in. And so protein kinase A is one of the three main metabolic kinases: protein kinase A, protein kinase B, and protein kinase C. Right, so we're going to talk about each of these three today. Sometimes they have different names, like protein kinase B is sometimes called AKT, but we'll see that when we talk about insulin signaling. Now, what is protein kinase A going to phosphorylate, right? Well, let's think about what epinephrine is actually signaling. It's signaling that energy is going to be needed because we're either going to need to fight or we're going to need to run away, right? So either way, we're going to need a lot of energy. So protein kinase A is going to phosphorylate targets, of course, right? It's the only thing that it can do. But in pathways where it produces energy, right? Targets of pKa are activated by phosphorylation. And in pathways where energy is stored, those enzymes that are targets of pKa are going to be inactivated by phosphorylation. So we are turning on the energy generating pathways and turning off the energy storing pathways just with phosphorylation, right? This is an example of what we call reciprocal regulation, which we'll revisit next week when we talk about glycolysis. All right, now epinephrine binding promotes cardiac muscle contraction in other ways as well. Okay, here, troponin forms a complex with myosin, okay, and it inhibits the ability of myosin to bind to actin. If you remember, myosin and actin, right, they kind of walk, or one walks along the other, okay? If we can't form that interaction where myosin walking on actin, well, what happens? Well, now we make muscle contraction more and more difficult. So troponin, which is normally present, right, prevents muscle contraction by inhibiting the interaction between myosin and actin, right? And these contain, of course, the troponin complex contains this protein here, troponin 1. Now, in cardiac muscle cells, protein kinase A, right, one of the targets is troponin 1, right? When troponin 1 is phosphorylated by protein kinase A, this weakens the interaction of troponin to actin, right? Phosphorylation weakens troponin binding to actin. So what happens now? Well, now if troponin is no longer binding to actin, this frees up myosin to bind to actin. So in this way, protein kinase A makes muscle contraction more easy in the cell by allowing myosin to bind more effectively to actin. Okay, we can see the whole pathway here. Epinephrine binding to the beta-adrenergic receptor in the heart specifically, right? Of course, this is going to lead to the activation of protein kinase here, right? Adenylate cyclase, increased cyclic A and P, activated protein kinase. Now, we saw already before that protein kinase A activation is going to lead to energy production, right? It's going to turn off energy storage, but also, right, protein kinase A is going to lead to enhanced interaction between actin and myosin, cardiac muscle protein phosphorylation, and better contraction, right? So protein kinase A varies in what it, well, it has different targets in different tissues because different tissues, of course, express different complements of enzymes or proteins, right? In the case of the heart, right, you express a lot of myosin and a lot of actin in order to promote muscle contraction. Now, we need a way to turn off these signals as well, right? We can imagine if we activated the beta-adrenergic receptor and then just left it on, well, then we'd be in fight or flight mode forever. So there has to be a way to turn off the signaling cascade for the beta-adrenergic receptor and for any receptor, right? In the case of the beta-adrenergic receptor, what we see is 4 different ways to turn it off. First, the G alpha S subunit turns itself off by hydrolyzing GTP, right? So we call these G proteins, okay, are almost always GTPases. G alpha is a GTPase. It will hydrolyze GTP to GDP. The kicker is that G alpha is actually very bad at doing this, right? So it takes a while. So in a way, kind of acting as a timer. How long it takes for it to hydrolyze GTP determines how long it will remain active, right? So after a certain amount of time, the GTPase activity of G alpha hydrolyzes GTP into GDP, and now we are no longer able to interact with adenylate cyclase. So adenylate cyclase is turned off, and then GDP-bound G alpha is free to reassociate with G beta gamma. These can now reassociate with either an active beta adrenergic receptor to be reactivated, or they remain inactive, right? So it turns itself off after a given amount of time. The receptor itself is turned off by the simple dissociation of epinephrine. Again, remember, no binding event is permanent, so after a certain amount of time, the right, epinephrine will dissociate from the receptor. Okay. Thirdly, the beta adrenergic receptor kinase, the GRK2 kinase, phosphorylates the C terminus, or the intracellular side of the beta adrenergic receptor. Okay, and what this does is it prohibits, or inhibits rather, the GNEF activity of the beta adrenergic receptor. Okay, and what is the GNEF activity? It's the guanine nucleotide exchange factor. And this GNEF activity is what promotes, we go back a few slides here, this is what promotes the exchange of GDP for GTP, guanine nucleotide exchange factor. This is promoted by the GNEF activity of the receptor itself. Right? So by phosphorylating the C terminus, the intracellular side, we inhibit that GNEF activity. And lastly, beta-arrestin recognizes these phosphorylated residues at the C terminus of the beta-AR receptor and further inhibit the GNEF activity so that it can no longer activate any inactive G alphas that it might encounter, right? So all of these lead to the inactivation of the receptor, okay? Now, after a while, right, beta-arrestin will fall off. These phosphorylated residues will be hydrolyzed, so we'll release the phosphate, and we're ready for another binding event, ready to respond to another epinephrine molecule. Now, there are other 7-TM receptors, obviously. So here, another way that we respond to epinephrine is the alpha-1 adrenergic receptor, or the alpha adrenergic receptor, and this leads to the activation of another metabolic kinase, PKC, right? But the way that this occurs is actually kind of a little bit more interesting than the way that the beta adrenergic receptor works. So not shown in this figure in your book for some reason is the actual alpha adrenergic receptor, but it's over here somewhere, right? Upon binding to epinephrine, or for example, angiotensin in this case, right? But here, upon binding to the alpha adrenergic receptor, this leads to the activation of G alpha Q, right? So this is a different type of G alpha. It's still a GTPase, right? But it's not the same as the G alpha that we saw in the beta adrenergic receptor. But this G alpha Q, right, leads to the activation not of adenylate cyclase, but instead phospholipase C, right? Phospholipase C is, as the name would imply, is a lipase, which means that it cleaves lipids, right? Specifically phospholipids. The phospholipid that's going to be acted on is phosphatidylinositol 4,5-bisphosphate, PIP2. Remember, right, from our lipids chapter, phosphatidylinositol is simply just a phospholipid. We have our glycerol backbone here with 2 fatty acids esterified to carbons 1 and 2, our phosphoryl group here, and our inositol alcohol here. PIP2 is simply phosphorylated inositol at carbons 4 and 5, okay? So, what happens now? Well, phospholipase C is going to cleave here, right? It's going to cleave, what do we call this? Phosphatidylinositol 4,5-bisphosphate is going to cleave it here between the glycerol and the phosphoryl group. So what are we left with? We're left with our inositol 4,5-bisphosphate with our phosphoryl group on carbon number 1 now. So what we're left with is inositol 1,4,5-trisphosphate, right? IP3, and that's what we see here. IP3 is cleaved from the phospholipid, phosphatidylinositol 4,5-bisphosphate, And this leaves us behind a glycerol with 2 fatty acids, or diacylglycerol, which behaves very much like a membrane lipid. In fact, it is a membrane lipid. So this remains in the membrane leaflet itself, free to diffuse laterally, again, like we saw in our membranes chapter. So now we have 2 secondary messengers, IP3 and diacylglycerol. We saw those on the 3rd slide or so. So let's focus on IP3 for now, right? What's going to happen to IP3 is that now it's free to diffuse in the cytosol, and it will be recognized by the IP3 receptor, which is present on the ER membrane, or the SR membrane in the muscle. And what happens upon binding to the IP3 receptor is that this calcium channel, it's a ligand-gated calcium channel, opens and allows the flow of calcium down its concentration gradient from the lumen of the ER to the cytosol. Remember when we talked about the circa pump, the endoplasmic reticulum is the storage compartment for calcium. So that's got a higher concentration of calcium than does the cytosol. Upon opening this ligand-gated channel then, just due to facilitated diffusion, calcium flows out into the cytosol. In the cytosol, right, it now makes its way over to protein kinase C, which is a peripheral membrane protein. When protein kinase C binds calcium, it is now partially activated, okay? It is fully activated by also binding diacylglycerol, which conveniently we also produced alongside IP3. So again, this diffuses laterally through this inner leaflet of the bilayer, binds to protein kinase C, fully activating it with the calcium. So now we have activated protein kinase C. Protein kinase C goes on to phosphorylate many downstream targets that regulate, for example, cell growth and other events. Okay? Now, calcium, when released into the cytosol, remember, we said when high amounts of calcium are present in the cytosol and in soluble precipitate forms, right? So we specifically don't want just a ton of calcium around in the cytosol. And in fact, we don't, right? So if we see down here, okay, we can measure over time, okay, calcium fronts, right? So essentially, how fast does calcium diffuse through the cytosol? And what we see is that calcium actually diffuses very slowly through the cytosol, much more slowly than you would expect for a small cation like calcium, right? So what's going on with calcium? Well, it turns out that calcium is never freely present in the cytosol, right? So here we're just looking at calcium using this Fura-2 dye, which simply just fluorescence in the presence of calcium. The higher the fluorescence, the more calcium there is, right? So orange means a lot of calcium, and green means very little calcium. So you can see that front of calcium moving through the cytosol. So why is calcium moving so slowly through the cytosol? It's because, again, we don't want all this free calcium in the cytosol. Instead, it's always immediately bound by this regulatory protein, calmodulin. Right? Calmodulin is made up of 4 EF hand motifs. And an EF hand motif is simply just 2 helices, okay, with a beta sheet in the middle, right? And if we look at the structure here of the EF hand, we can see that calcium is in coordinated bonds with many side chain and main chain oxygens, right? So calcium is always, like any metal ion. We saw this with iron in hemoglobin, right? It's always going to be coordinated to something. In this case, it's coordinated to all these oxygens, anywhere between 6 and 8 oxygens. So it's being held in these EF hand motifs. And calmodulin is made up of 4 EF hand motifs, each one of which can bind to 1 calcium. So a calmodulin is capable of binding 4 calcium cations. Okay? Now, what is the importance of calmodulin? Well, when calmodulin binds these calciums, right, because it can form all of these coordinate bonds, it can change the structure of calmodulin, right? It can stabilize it into its active form, okay? So here we have, for example, some calmodulin activated kinase, right? So some kinase that's activated by calmodulin. This has got a calmodulin target peptide. So what happens here is that when calmodulin binds its 4 calciums, it changes its conformation so that it can bind this CAM target peptide, that calmodulin target peptide, on our calmodulin activated kinase, okay? Converting this inactive form into its active form when it's bound to activated calmodulin. That's what's going on here with protein kinase C. The calcium leaves the ER lumen, binds calmodulin, the calmodulin recognizes CAM peptides in protein kinase C, activating it alongside again with that diacylglycerol, leading to downstream targets. Yes.
I just don't get the hand. Why is it the hand? You have hand pen.
Oh, because it kind of looks like a hand.
Oh.
So in the first protein that was identified to bind calcium had many alpha helices, and typically these alpha helices in these small proteins, like in myoglobin, are lettered A, B, C, D, E, F, G. It just so happened that in this first protein, where the calmodulin bound was in helices E and F.
So where the fingers are pointing us like the direction of the helices?
Yeah, it just kind of looks like a hand, right? So it's a thumb and the blue helix and the index finger and the yellow helix. There's nothing. Don't worry about the hand. That's just what it looks like. Okay. But it just so happened that, right, so that it was found in helices E and F in that specific protein, but what they saw was this same pattern of helix, beta sheet, helix in other proteins that bound calcium, right? So they just called it the EF hand because that's the first one that was found. All right, some receptor signal through G proteins that inhibit adenylate cyclase, right? So not only activate adenylate cyclase, but sometimes we inhibit adenylate cyclase. So in the case of the alpha-2 adrenergic receptor, which is not to be confused with the alpha-1 adrenergic receptor, the alpha-2 adrenergic receptor is again a 17M receptor, but signals through G-alpha-I, right? So not G-alpha-S that we talked about in the beta-2 adrenergic receptor, or G-alpha-Q in the alpha-1 adrenergic receptor. The alpha-2 adrenergic receptor signals to G-alpha-I. This G protein binds and inhibits adenylate cyclase, right? So this is actually expressed in some smooth muscle cells and in blood vessels. So they're very much similar in that way. It causes blood vessels to constrict, and it causes smooth muscle to contract, right? So in this case, right, we are activating the receptor, but in this case we're activating G-alpha-I, which inhibits adenylate cyclase, and this of course is going to lead to decreased cyclic AMP levels, because far from making more cyclic AMP, we're allowing cyclic AMP to be hydrolyzed by cyclic AMP phosphodiesterase, generating just AMP, which is not going to activate anything. And this causes a decrease effectively in the amount of active protein kinase A. And in smooth muscle cells, decrease in protein kinase A actually leads to contraction, right? And in blood vessels, which are very similar structurally to smooth muscle, causes constriction as well, right? And of course, these are all responding to epinephrine. So we see that epinephrine can have opposite effects depending on the tissue type, right? So here we've been talking about epinephrine with reference to, not smooth muscle, so like heart cells, okay? So in this case, we were trying to generate energy by activating adenylate cyclase, activating protein kinase A, activating protein kinase C, and all this kind of thing. But in other tissues, we want to respond to epinephrine in different ways. Now, of course, nothing's changing about epinephrine, okay? So instead, what we do is we just don't express the beta-2 or the alpha-1 adrenergic receptor. Instead, we express the alpha-2 adrenergic receptor, right? So there's a different complement of receptors that might all recognize the same primary messenger, but have different downstream effects. Okay, epinephrine binding promotes cardiac muscle contraction. We're going to talk about this. So the adenosine A1 pathway actually is how caffeine works, right? So the adenosine A1 receptor signals through G-alpha-I. Okay, so this is another example, like the alpha-2 adrenergic receptor. Here, the adenosine A1 receptor signals through G-alpha-I. And in the muscle cell, right, this has a different effect, obviously, than it would in blood vessels or in smooth muscle. So what's going to happen? Well, let's imagine what happens when adenosine is present then. Adenosine present in the heart is going to inhibit adenylate cyclase, so it's going to inhibit the amount of active protein kinase A, right? And remember, protein kinase A is going to, or active protein kinase A rather, is going to increase the amount of myosin that can bind to actin, and it's going to increase the amount of energy available to contract the muscle cells. Now, if instead we are signaling via G alpha I due to the presence of adenosine, well, now this is going to decrease the amount of protein kinase A, which is going to decrease both the amount of energy and the effectiveness of muscle contraction due to the reduced ability for myosin to bind actin. So this will slow the heartbeat due to a decreased amount of cyclic AMP, and it's going to decrease the release of certain neurotransmitters that we're not going to get into right now. But just in terms of what we've talked about already today, right? It's going to slow the heartbeat because there's less energy and there is less interaction between myosin and actin. Here it's acting actually in the brain to reduce, the amount of times it's telling the heart to beat. The antagonists for this adenosine A1 receptor, like caffeine, well, what effect will they have? Well, we talked about caffeine as a competitive inhibitor for the adenosine receptor. We talked about enzyme kinetics. Okay, so what's going to happen here? The Vmax of the receptor remains the same, right? But the Km decreases, which means that we would need more adenosine to achieve the same result, which means that at the same level of adenosine, okay, we will have less activity, which means less G alpha I will be activated, which means that less inhibition of adenylate cyclase, which means that the activity of protein kinase A will remain a little bit higher, right? So, you ever get the jitters when you drink too much caffeine, right? That's why, because you are inhibiting the ability of adenosine to inactivate adenylate cyclase, right? So this leads to high heart rate and stronger heartbeats, right? So you get really jittery to the point where caffeine is actually lethal if you drink too much, though it's very hot. It is theoretically possible. I suppose it's theoretically possible to die of drinking too much water also, but I guess everything is a poison if we drink enough. Okay, so onto the insulin receptors. So insulin receptors and epidermal growth factor receptors are a different class entirely of receptors. They are not 7-TM receptors. Instead, they belong to a family of receptors called receptor tyrosine kinases. If you remember the structure of tyrosine, okay, it can also be phosphorylated. It has a aromatic group, right? But it's got a hydroxyl at the end. So it's like phenylalanine, but with a hydroxyl at the end. This hydroxyl can be phosphorylated. Oh, here's tyrosine right here. Okay, so we can form phosphotyrosine. And in fact, that's exactly what the insulin receptor does. So the insulin receptor exists as this dimer in its inactive state. Okay, on the inside, right? So here we have the outside of the cell. Here we have the inside. On the outside, obviously, is where we're going to bind insulin. On the inside is where we have our beta subunits or the tyrosine kinases. And what is going to happen is when insulin binds to the outside, right, these activation loops are going to move in such a way where now this beta subunit of the insulin receptor can phosphorylate tyrosines on the opposite one, and this tyrosine kinase can phosphorylate tyrosines on this opposite one. So we get cross-phosphorylation between the beta subunits. As we see here, we combine up to 4 insulins, and this changes the conformation of the insulin receptor, right? Bringing these two closer together, the 2 beta subunits, but now they can act on each other, and tyrosine residues are phosphorylated on either one of the beta subunits by the opposite beta subunits, and now we have something that initiates our signaling cascade, because these phosphorylated tyrosine residues that we can see here are going to be recognized by IRSs, insulin receptor substrates. So for example, the first IRS, IRS1, contains what we call an SH2 domain. And an SH2 domain is simply just something that recognizes a phosphotyrosine, right? So when you see an SH2 domain, you just know that it recognizes a phosphotyrosine. And that's what we have in IRS1. When this recognizes the phosphotyrosine, the tyrosine kinase activity of the beta subunits also phosphorylate tyrosine residues in IRS1 itself, right? So now these tyrosines on IRS1 are phosphorylated, and IRS1 is also held to the inner leaflet, right, close to the receptor by this domain here, which recognizes PIP2, the same PIP2 that we saw in the PKC cascade. Now, these phosphotyrosines on IRS1 are recognized by phosphoinositide 3 kinase, right? So let's look at the name. We know it's a kinase. It's going to phosphorylate something. What it's going to phosphorylate is phosphoinositide at position 3. So we turn our PIP2 into PIP3, phosphatidylinositol 3,4,5 trisphosphates. Okay, and again, this is recruited by its SH2 domain binding to the phosphotyrosine on IRS1. So now we have our PIP3, which now goes on to activate PDK1, PIP3 dependent protein kinase, is activated again by PIP3. This in turn then activates protein kinase B, right? So this PDK1 activates AKT, which is PKB, the third metabolic enzyme. So when we talk about glycogen synthesis in chapter 21, what we're going to see is that protein kinase A, which is activated in response to glucagon, is going to have an opposite effect as protein kinase B, which is activated in response to insulin, right? So another example of reciprocal regulation. Insulin, right, is going to increase the amount of glucose uptake and glucose storage, okay, whereas glucagon, which is released in times of starvation, is going to inhibit glucose uptake and instead mobilize glucose to be used by the cell, right? So opposite effects. Here's the same thing, right? So we have several levels of amplification here, okay? The receptor is activated, right? Which can lead to many phosphorylated IRS proteins. So there's our first level of amplification. These phosphorylated IRS proteins each bind phosphoinositide 3 kinase in a 1-to-1 ratio, right? So there's no amplification here. These phosphoinositide 3 kinases can phosphorylate many PIP2 molecules. So there's our second level of amplification, right? Phosphatidylinositol 3,4,5 trisphosphate activate one PDK1 in a stoichiometric ratio, so there's no amplification there, but now PDK1 can activate many PKBs, right? So we have 3 levels of amplification there, just like we did in our beta adrenergic receptor. I'm gonna skip epidermal growth factor because I do want to get to vision and smell, and I don't know how much longer I have standing energy. Okay, the EGF pathway, all right, skipping. So epidermal growth factor you don't have to worry about. It's not going to be on the exam. We just touched briefly on it. It's another receptor tyrosine kinase. Okay, so sensory systems are based on signal transduction pathways, right? So I do want to talk about these, and then we'll call a little bit early today, if that's all right with everyone. I'm sure everyone wants to get out on Friday. Yeah, me too. Okay, go take a nap. Fucking kids, man. They come, they, you know, I don't get it. They just lick shit all the time. They just have no awareness of germ theory. They just sneezing on, like, whatever. Ooh, okay. So, sensory systems. All right, so let's start with odorants. So odorants are small organic compounds, obviously with, importantly, volatility. So they have to be able to be in the air for you to smell them. Okay, so we have things like benzaldehyde, 3-methylbutane, 1-thiol, which is what skunk smells like. I'm sure everybody smells a skunk driving around New Jersey. Rose, spearmint, caraway, ginger, right? So we can break down specific smells and correspond them with specific molecules, right? Importantly, they're all small molecules, and in order for us to register them in our brains, there has to be some, lock and key. There has to be some receptor. In fact, there is, and these are called olfactory receptors, which are linked to olfactory neurons. And these look suspiciously similar to the beta adrenergic cascade. In fact, they're almost identical. So we have some, olfactory receptor here. This is going to be specific for one odorant, right? And we have about 400 different types of olfactory receptors, all right? And each olfactory receptor binds its respective odorant, but then it works exactly the same, okay? All the olfactory receptors work exactly the same. They activate G-olf, right? G-OLF, which is similar to G-alpha, a little bit different, okay? But here, this dissociates from G-beta gamma like we've seen before. G-OLF, which is now active, right, due to the GNEF activity of this receptor, exchanges its GDP for GTP, is now active. It associates with adenylate cyclase, which again converts ATP into cyclic AMP, but now in specifically these receptor cells in our nose, they don't activate protein kinase A. Instead, what they do is they open these non-specific cation channels, allowing cations to move into the cell, okay? And these now, for example, sodium and potassium, or sodium and calcium rather, these flow into the cell. And what did we find out last Tuesday, right? Whenever we open an ion-specific channel, we depolarize the membrane. This leads to the production of an action potential, and this action potential then rushes up into our olfactory bulb in our brain, okay? And we have a connection, a one-to-one connection between a receptor cell, a receptor neuron, and somewhere in our olfactory bulb. Dr. Monahan, third floor of Nelson, he works on exactly this. I was talking to him the other day, and our smell, I always thought our smell was not that good compared to dogs, but apparently we are able to distinguish, you know, pretty much the same smells. It's just that they have more receptors, right? And the olfactory bulb is also one of the only brain structures that don't scale with size, right? So, you know, if you think about a whale brain, it can be quite large compared to ours, but the olfactory bulb is not any bigger necessarily, right? Because it scales with the number of neurons, right? With the number of receptors that are expressed, not with the size of the end, right? Because we have a one-to-one connection between these neurons and the olfactory bulb. Right, so now we have, you know, about 400 of these different types of receptors present in a certain number of copies, right? So we don't have only 400 cells sensing this. We have many thousands, but overall we express 400 different types of receptors, so we can distinguish between 400 individual odorant molecules, right? So depending on which cell, right, and each cell only expresses, by the way, one specific type of receptor, so depending on which cell generated the action potential, our olfactory bulb was able to tell, well, what type of odorant must have been present in order to generate that signal. So that's how we can tell smells apart, okay? Now, we express, again, many different types of odorant receptors, right? Or actually over here, receptors. So here's just showing 14 receptors, but each odorant molecule can activate several different odorant receptors. Okay, so yes, we have ones that are specific to specific odorants, okay, but we can get crosstalk between them, right? So let's see here, this 8 carbons with a carboxylic acid at the end, all right, this is going to trigger multiple receptors. So depending on which cells generate the action potential, our brain puts together which combination of cells led to the production of action potentials, right? And then tells us, oh, this is, you know, rosemary, or this is ginger, or whatever, right? So it's a combination of receptors all working together, right? So this is why we have to learn our smells, because we're, you know, during development, we're still learning how to put together these signals and express them as, oh, that's this specific smell, or that specific smell, right? Vision works very much the same way, right? So vision relies on specialized 17 receptors and responds not to molecules, but instead to electromagnetic radiation, right? So we have the entire wavelength, the spectrum here, but the visible spectrum is actually quite small, going wavelength only from about 10 to the minus 7 to 10 to the minus 6 meters, right? So about 1 micrometer, so 10, whatever. Can't do the math right now, so you can do the math. So vision is based again on the absorption of light, and we have 2 specific types of photoreceptors. They work more or less the same way. I'm going to focus on cones, right? Cones are responsible for, or no, sorry, rods are responsible for just general light level, right? So they generate an on or off signal, but our brain doesn't interpret that as color. It just interprets that as the presence of light. All right, so I'm going to focus on rods. Cones, however, we have 3 different types of cones. One's that's specifically receptive to red light, green light and blue light is where we get the RGB from computers, because they stimulate physically different cones in our eyes, but they function very much the same way. So I'm just going to focus on rods. So rods are called rods because they resemble rods, and they're made up of these discs here, right? These is a neuron, right? So this is going to generate some action potential, which again migrates back, so our eyes are connected directly to our visual cortex, right? So there has to be some kind of action potential telling the visual cortex, oh, this neuron just triggered, or that neuron, or this rod just triggered, that rod just triggered. And then we put together all that in our heads. Again, we have to learn how to do this. Babies are functionally blind. Aside from the fact that their muscles aren't able to focus, their corneas, they're still learning how to process, frankly, a massive amount of data, right? So if you imagine your eyes are constantly generating action potential, even when your eyes are closed, right? You're generating action potentials that are constantly being processed by your visual cortex, which is one of the theories behind deja vu, actually, is that sometimes your visual cortex gets overwhelmed, right? So it processes things a little bit slower, but it's already been sent to your short-term memory, so by the time your visual cortex processes it, remember that you've already done this, but it's just a quirk of your brain, because our brains are too complex for our own goods. All right, so the special secret sauce here in rods is this molecule here, 11-cis-retinol. And 11-cis-retinol is actually in a shift-based linkage with a specific glycine and a protein called opsin, right? So that's where we get the opsin in rhodopsin. And 11-cis-retinol does something kind of weird when it encounters a photon. It changes from the cis configuration into the trans configuration. And when we change into the trans configuration, it actually moves this nitrogen on the lysine of opsin and moves about 5 angstroms up, right? So when we convert cis-retinol into trans-retinol, again, we get this very significant 5 angstrom movement of the nitrogen in lysine, right? So what happens is that when this this trans rhodopsin here, or retinol rather, sorry, I got my name confused. When this trans retinol moves that nitrogen, this activates this G-alpha protein called transducin. Okay, transducin also generates an action potential, but in a different way than G-alpha did, for example. So again, this activates the GNH activity of rhodopsin. It activates transducin, so it exchanges the GDP for GTP. This activated transducin now doesn't interact with adenylate cyclase at all. Instead, what it does is it activates a cyclic GMP-specific phosphodiesterase. And what it does is it hydrolyzes cyclic GMP into GMP. So this removes cyclic GMP from this ligand-gated channel, which is typically allowing sodium and calcium in, right? So now when we remove cyclic GMP, this inhibits, it closes this channel and generates an action potential that way, right? So we close the channel and we generate an action potential, depending on which rod cell generated the action potential, right? So again, there's a one-to-one mapping between rod cell's position and our visual cortex. We can tell which rod cell was activated, which generated the action potential, and then we put that together as an image, right? This black and white image, or in low light. Cones work very much the same way. They're a little bit more complicated because they don't have to respond to just photons in general. They have to respond specifically to photons of a certain wavelength, right? But they lead also to the generation of action potential through, again, one-to-one map in our visual cortex. And then what our visual cortex does is it puts together the light information, right? And superimposes that with the images that we get from the red, green, and blue cones to put together a whole image. And this is happening continuously. Right? It's not like on the computer or on the TV where you can have 30 frames per second or 60 frames per second, right? This is happening continuously. I think the limit of we can perceive is something over 100 frames per second. Somebody who's into video games might know more than me. Anyway, hearing is also mediated by these types of signal transduction pathways. So in our ear, we have these stereocilia, okay? And what these do, the tips of these stereocilia, which are essentially just hairs, okay, are coupled to ion channels, okay? And these are gated not by light or by a ligand in the case of cones and GO. Instead, they're gated by mechanical action, right? So mechanical action actually activates these channels, right? So here again we have these stereocilia, they're linked together at their tips. So when a wave, the sound wave comes in, right, it physically moves these, right? So if you've ever been at a concert or whatever and you stand too close to the speaker, I don't know nobody's done this, but you stand too close to the speaker and you feel your hair moving, it's the same thing. That's a very loud speaker, right? But we have much more sensitive and tinier hairs in our ears. It's the same thing. They're moving, and when they move, okay, this causes a displacement which activates these mechanoreceptors. These mechanoreceptors open again gated channels, but in this case these gated channels are responding to mechanical stress. And this generates an action potential, which then again goes to our brain, and we interpret that as sound. So all our senses really are just our brain's interpretations of electrical signals generated by these signaling cascades, right? So I don't get too esoteric here, but we're all just, you know, electrical machines kind of guesstimating what reality is really like. All we're doing is just responding and trying to make sense of this frankly overwhelming amount of data that's coming into our brain at any given time, right? So that's signal transduction, very interesting stuff. Next week, all right, we are moving on to metabolism, right? So we're going to start with an overview of metabolism, some general principles and concepts, and then we get into the really fun stuff like glycolysis and the electron transport chain. All right, so I'll see you all next week. Have a good weekend.